So now that we know the structure of a membrane, we can look at how it controls what comes in and what goes out of a cell. A cell needs many different molecules to operate effectively, and it also needs to get rid of waste. So in this section, we'll look at the various methods for moving molecules in and out of cells. So in this lesson, we're going to cover passive transport. Passive transport means it doesn't require energy, and this includes diffusion, facilitated diffusion, and osmosis. We're also going to try and explain how the properties of molecules affect how well they are transported, their size, their charge, their solubility. We're going to look at the process of active transport, which requires ATP. And we're also going to learn a little bit more about ATP and how it is used, how the hydrolysis of ATP is used to provide that energy. We're going to look at something called bulk transport with endocytosis and exocytosis, where we want to move large amounts of stuff in or out of a cell. So diffusion. Now diffusion you will have covered before. It's the simple movement of particles in a liquid or a gas from a high concentration to a low concentration, or down a concentration gradient. Now small uncharged molecules can pass between the phospholipids by diffusion. So things like carbon dioxide and oxygen can pass no problem between the phospholipids. Water is small, but it does have a little bit of a charge. Remember, it's a polar molecule, but not enough to stop it moving, but it's not that efficient, okay? But water can pass through the phospholipids. Glucose, too large, won't pass between the phospholipid bilayer. And also charged ions, things like sodium and chloride ions, will not be able to pass through either. Okay, diffusion, it's a passive process, doesn't require energy, just relies on the random kinetic movement of particles. The rate of diffusion is affected by the concentration gradient, the difference between one side and the other, the area over which diffusion takes place, the distance over which diffusion takes place, and the temperature and therefore the kinetic energy of the particles. And you should be able to explain those and be able to interpret graphs in an exam question using your knowledge about those factors. So, facilitated diffusion. Now this is diffusion but with the help of proteins. There are two types of proteins involved here, channel proteins and carrier proteins. Channel proteins are going to be able to allow things like ions through. This is a sodium channel here, specifically allows sodium through. And then you've got carrier proteins which allow large molecules to pass through. It's still a passive process. Molecules are still passing from a high concentration to a low concentration. They are just using these proteins to get through the membrane because they couldn't get through the phospholipids beforehand. Carrier proteins do change shape, but it doesn't require any energy for that process to happen in this case, but they are used for larger molecules. Like I said, they are specific. So this one is a glucose carrier protein here. This is a graph that you should be familiar with. As you can see, you've got external concentration of glucose, which is increasing, so we're getting more and more glucose building up on one side of the membrane, and you'd expect, therefore, the rate to continue to increase, the rate of diffusion to continue to increase, which it does, but then it levels off. But that's because this is facilitated diffusion. The carrier proteins get saturated. There's only so many of them, and so if you keep the concentration going up, at some point they're all busy, and you can't make the process go any faster, so it does plateau off. Okay, osmosis. You will have covered osmosis before, but we're going to go into a little bit more detail in A level. Osmosis is the diffusion of water. It's the net movement of water molecules through a partially permeable membrane from a region of high water potential to a region of lower water potential. High water potential is another name for a dilute solution, and low water potential is more of a concentrated solution, more stuff dissolved in it. We measure water potential in kilopascals, kPa, and it has the following symbol. Now pure water, the highest water potential possible, actually has a water potential of 0. And then the more stuff you dissolve in it, the lower the water potential becomes. So you go into negative numbers the more you dissolve into that solution. Here we've got a membrane, we've got a solution either side, we've got a high water potential on the right and a low water potential on the left. Now the idea of water potential is how much free water there is that has the potential to move. On the right, every water molecule there has the potential to move. On the left of the membrane, there are some sugar molecules which are interacting with the water molecules in order for the sugar to be dissolved. And that is kind of occupying some of those water molecules. Those ones couldn't move anymore because they're interacting with the sugar. So there's less free water, so it is a lower water potential. Therefore, water moves from the right to the left in this situation. In this situation here, we've got the opposite. We've got a high water potential on the left and a low water potential on the right, caused by the salt dissolve there. The water will move onto the right-hand side to balance the water potential, but you can see the volume will actually shift because of the volume moving. You can also talk about osmosis in terms of tonicity. Tonicity is how much solute is dissolved in the water. Okay, so if a solution is hypertonic, that means lots of solute is dissolved in it. Hyper, lots of tonic. Okay, hypotonic, not very much solute dissolved in it. Okay. And isotonic, if you have two solutions that are isotonic, it means they are balanced, they are the same tonicity. So you want to have isotonic solutions to put cells into. So if you're having a drip in a hospital, that will actually be a 0.9% sodium chloride solution to make sure it's isotonic to the water potential of your blood cells so they don't burst. So if you take animal cells and put them in different solutions, a hypertonic solution will cause water to move out and the cells to shrink or become created. Hypotonic will cause water to move in, the cells will burst, what we call lysis. Plant cells behave a little bit differently because of the cell wall. A normal plant cell in isotonic solution would be slightly flaccid. Water hasn't lost loads of water, but also it's not gained loads of water to push out on the membrane. If it gains lots of water in hypertonic solution, that's turgid. If it loses lots of water, it may become plasmoid, where the membrane peels away from the cell wall like that. So here's a situation, we've got a low water potential inside and a high water potential outside. These are showing, the red and white dots are showing water molecules, H2O. So we've got more water outside than inside. Water wants to move in. It will move in by osmosis, okay, from a high water potential to a low water potential. The volume inside the cell, though, at this point then expands and starts pressing against the cell wall with a force called turgor pressure. This resists the further uptake of water, and this is known as the pressure potential. And this is always a positive value. It's always pushing out, trying to force water out, so it's always positive. The dissolved solutes inside create a solute potential trying to draw water in, high water potential, low water potential. Therefore, you can think about the total water potential as a combination between these two potentials. The solute potential drawing water in and the pressure potential pushing water out. And we can see it in this equation here. So sometimes we're quite interested in finding out about the water potential of a plant cell. And we want to find that point at which there isn't any pressure potential being put on the cell wall when it's zero, because when the pressure potential is zero, then the water potential has to be equal to the solute potential, which we know or we can work out quite easily. This point is called incipient plasmolysis. Now, we can't just observe that by looking at a cell. We can't tell if the pressure potential is zero. But what we can do is we can observe a whole load of cells and we can see how many of them are plasmolyzed and how many of them are not plasmolyzed. And if we pick the 50% plasmolysis point, where 50% of them are plasmolyzed, 50% are not plasmolyzed, on average, all the cells are at this incipient plasmolysis point. Okay, active transport. Again, you probably have heard of this before. It's when you want to move something against a concentration gradient. When you want to do that, that's going to require energy because you're going against what diffusion is naturally wanting to occur. Okay, so we're going to need energy. Where do we get the energy from? We get it from ATP. So here's a glucose molecule. We actually want it to move in to the cytoplasm here from a low concentration to a high concentration. So we're going to need a protein pump for this in the membrane. So it's another carrier protein, but this one specifically is a pumped protein that uses ATP, which gets converted to ADP and phosphate. That releases some energy, which is enough to move that glucose molecule from a low concentration to a high concentration. As I said, the energy provided comes from ATP, and it's when you perform hydrolysis on ATP and split it into ADP and phosphate that energy gets released. And there is an enzyme that does that hydrolysis of ATP. It's called ATPase. This is a nice example that you'll come across later on in the course when you're studying the nervous system of active transport. It's the sodium-potassium pump. It's a very special protein pump. It pumps 3 sodium ions out of the cell and 2 potassiums in at the same time using ATP, active transport. The potassium ions flow back out and the process reoccurs, and it keeps going until you get a complete difference in gradients between sodiums outside and potassiums inside. And this is all very important in generating an action potential in a nervous impulse, which you'll learn about when you study the nervous system. Now you need to know a little bit about the evidence for active transport and the evidence that ATP is involved. Well, basically, the rate of active transport is affected by oxygen concentration and temperature. Now, oxygen concentration and temperature affect the rate of respiration where ATP is produced. So that suggests that you need ATP for active transport to occur. Also, poisons that stop ATPase from working, such as cyanide, seem to stop active transport. And then if you put some ATP artificially back in, the whole thing starts up again. So that would suggest that you need ATP for active transport. Our final type of transport is bulk transport. This is when you want to move more than just one molecule. You want to move a whole load of molecules in one go, okay, or really large molecules. Sometimes the cell needs to secrete loads of waste, or sometimes it needs to draw a whole load of stuff in. Bulk transport requires ATP. It is a form of active transport. Endocytosis is when you move a whole load of stuff into the cell, and exocytosis is when you move a whole load of stuff out of the cell. The reason this is possible because of the fluid nature of the phospholipid bilayer, where you can just pinch off bits of membrane and add bits of membrane back in. Now, endocytosis can be split up into two further processes. Phagocytosis is when you're taking in large whole molecules, even a whole organism. It's basically what we call cell eating, and the vesicle that's formed is large. It's 250 nanometers. Okay, an example of this is phagocytes, when they engulf prokaryotes, white blood cells engulf these bacteria. Pinocytosis is when you small vesicles. It's also referred to as cell drinking. Okay, and this is carried out by all eukaryotic cells. Exocytosis, releasing stuff out. Okay, like I said, quite often things like secretion of a hormone out of an endocrine cell. So here's a summary table showing you all the types of transport you need to know about.